ऑक्सिन, ओके। ऑक्सिन जैसा हमने पहले डिस्कस करा, इट ग्रीक मीन्स, comes from the Greek word oxein, which means to grow. So there was a story, how was it discovered? Okay, so discovered जो हुआ था, it was discovered by two persons, father son duo, that is Charles Darwin and उनके son जो थे Francis Darwin. What they observed was जो कोलियोप्टाइल है of a plant, that is canary grass का जो कोलियोप्टाइल था, when it was placed in the path of light, okay, yeah, let us say कि इस side से we are getting light, so what will the plant do is the plant will show movement in the direction of light. So this plant, this was the coleoptile, and this is the canary grass on which they observed this thing. So light के direction में there was a supposedly bending of the coleoptile or the tip portion of the plant of canary grass. So they thought कि there must be some chemical influences working here. So उसे जानने के लिए what they did was they did a simple experiment कि उन्होंने जो टिप था, they thought that probably इस टिप में ही कोई केमिकल रिलीज होता होगा जिसकी वजह से it's bending or not. So उन्होंने क्या करा कि they removed the tip. उसके बाद जो रिजल्ट हुआ, the result was this कि the stump or the remaining portion did not bend at all. So इससे there was a confirmation, observation ये था कि it is the tip and the tip only जिस पे कोई chemical produce हो रहा है. They didn't have any idea कि what chemical is produced, but कोई chemical produce हो रहा है which is responsible for bending. और जैसे ही उन्होंने tip remove करा, there was no bending motion seen. So what they did, they again placed the tip back here. उसका result ये हुआ कि now the plant again showed movement. So there was some sort of let's say chemical influence which was traveling from the tip up to this portion, and it is this portion, not the tip, which is showing you the movement or the bending towards light. Or there was they thought probably कि let's check for another stimulus. So what they did, they took the coleoptile and they covered it with a foil. Should that bend? Unfortunately, that did not bend. So they came to the conclusion that light is being the stimulus here, जिसकी वजह से there are some sort of chemicals that are released, और वही chemicals are responsible for the bending. Okay, so now how did they discover कि what is the chemical? उसकी जो discovery हुई थी, it was extracted from, okay, let's write the scientist name, that is Charles Darwin and his son Francis Darwin. Okay, Charles Darwin and Francis Darwin were the discoverer of this plant hormone, and उसे extract कहाँ से किया गया था? The extraction that was done in this case was from ह्यूमन यूरिन, ओके, क्योंकि ह्यूमन डज नॉट हैव एनी receptor or any use of a plant hormone, so when we eat the plant, these hormones are present in our urine because they're not metabolized in the body. So uski wajah se human urine mein this was found and this was extracted for the first time. Now auxin ke there are two classes of auxin. The first class is natural sources, that is the one जो plant में produce हो रहा है, and they are two in number, IAA and IBA. okay, IAA का full form है indole acetic acid. okay, indole acetic acid, while IBA stands for indole 3 acetic acid and there is 3 butyric acid. okay, now artificial sources से भी बनते हैं, and the artificial ones are 2,4D and 2,4,5D. These are the artificial sources of auxin. So now extraction जब हुआ था, there was another scientist who is mentioned noteworthy here, and he is W.H. Wendt. W.H. Wendt ने कोलियोप्टाइल के जो टिप्स थे, कोलियोप्टाइल के टिप्स से he extracted the hormones. And how did he do that? He did a very simple thing, कि he took the tip और उसको separate कर दिया. Let's say this is the tip and this is the whole plant, whole cannery grass. So what he did was he took the tip and he separated it from the rest of the plant. Next, इसी tip को he took and placed it on an agar block. Now agar में he allowed this chemical जो इसमें था, that is auxin in this case, to diffuse into the agar block. As soon as it diffused into the agar block, उसके साथ ही उस agar block में सारे auxin के जो molecules थे वो concentrate हो गए. And next, जब उसने tip के जगह दोबारा auxin block को लेके उस plant पे place करा and light वाली stimulus दी, the plant bended. तो उसका ये मतलब है कि जो chemical ingredient था, ये जो सबसे important यहाँ पे hormone था, that diffused from the tip of the coleoptile into the next one, into the agar block. Extraction of auxin from coleoptile tip using agar. Okay, so that is it. Now how do we know what effect does auxin have on the growth of a plant and all? So uske liye we do a test which is known as bioassay. Bioassays are important क्योंकि इससे हमें एक particular chemical substance, एक particular factor को test करने का मौका मिलता है, जिससे हम बता सकते हैं what role, and अगर role है तो in how much quantity एक chemical या let's say एक phytohormone हमें चाहिए plant growth के regulation के लिए. There are two tests in the bioassay में for auxin. That is, first one is Avena curvature test. And the second one is root growth inhibition test. ओके, जो ऑक्सीन है, ऑक्सीन इज रिस्पॉन्सिबल फॉर द डेवलपमेंट ऑफ रूट्स पर्टिकुलरली. सो अगर ऑक्सीन की क्वांटिटी बहुत ही कम है इन अ रूटिंग मीडियम, so the plant will not develop roots properly. So by that method हम deduce कर सकते हैं कि कितने concentration of oxygen is required for the proper growth of roots or the development of roots. Now we will talk about its physiological aspects. The physiological aspects, okay, physiological aspects. Physiological aspects, okay. The first one is abscission. The second one is bolting. Sorry, its bolting is not seen here. This is apical dominance. The third one is cell division. And cell division and let's say xylem differentiation. And the fourth one is so let's talk about individually what do they mean. So abscission का मतलब we had talked about earlier, abscise or to cut or something. So abscission is a phenomenon जिससे plant leaf, fruit and all they fall off. So what auxin does is auxin prevents it. That is, वो प्लांट के जो भी लीफ है, फ्रूट है, दे इट विल डिले सेनेसेंस फॉर इन दिस केस, और इट विल नॉट अलाउ द प्लांट पार्ट्स टू फॉल ऑफ. सेकेंड इज एपिकल डोमिनेंस. नाउ हमने जैसे पहले एक्सपेरिमेंट में देखा था कि इट इज द टिप जहां पे ऑक्सीजन इज प्रोड्यूस्ड. सो इस टिप पे ही ऑक्सीजन प्रोड्यूस हो रहा है. सो व्हाट हैपेंस इज कि जैसे ही लेट्स से वी हैव अ प्लांट हियर, दिस इज द प्लांट टिप, दिस इज द लीफ, this is the leaf, okay, and यहाँ पे there are buds. Buds is let's say a condensed shoot or branches. So this is our apical bud, this is a lateral bud. So what happens in the influence of the apical bud is that apical bud में there is lot of secretion of auxin. Okay, so जैसे auxin का secretion होगा, it will stop the growth of the growth or development of the lateral buds. इसका effect ये होगा कि the plant will continue growing in length. Okay, लेकिन उसकी जो growth in the form of lateral growth नहीं होगी. The plant will only grow taller and taller. So what happens is, अगर हमने let's say apical bud हटा दिया, so जैसे ही हमने apical bud हटाया, the plant will not grow that tall जैसे वो उसके presence में कर रहा था, but the plant will grow a lot of let's say lateral buds develop हो जाएंगे. इन शॉर्ट जो प्लांट है, इंस्टेड ऑफ ग्रोइंग टॉलर इट विल ग्रो बुशियर. नाउ ये जो टेक्निक, दिस प्रोसेस ऑफ रिमूवल इज यूजफुल इन सर्टेन प्लांटेशंस, पर्टिकुलरली टी, जैसे टी प्लांटेशंस होते हैं. टी प्लांटेशंस में इट इज वेरी इंपॉर्टेंट बिकॉज जैसे ही you remove the apical buds, you'll have a rapid growth of the lateral buds. और lateral buds जैसे ही ग्रो करेंगे, लेटर बड के ग्रो करने के साथ ही the number of new leaves that are coming out they will also increase. तो इससे profitability increase हो जाएगी और production भी increase हो जाएगा. So prevents abscission, prevents this is abscission of younger parts of the plant. Okay, apical dominance increases length, decreases lateral growth. And the last one is cell division and differentiation. Now cell division is a normal process, लेकिन cell division का role जो होता है growth में, it's very important. So auxin what it does, is that it promotes cell division और फिर xylem differentiation. So in short, it is preparing the plant for a greater phase of growth. So cell division is shown by this and another hormone, plant hormone, that is known as cytokinin. So these two are synergistic. Okay, these two are synergistic. We'll talk about synergistic and antagonistic in the succeeding part. Synergistic phytohormones. Okay. So rooting medium में if we have let's say we are developing or culturing plant cells, so if we want to induce the development of roots, तो हमें high concentration of auxin चाहिए. This will help to induce the roots in the plant, root development. In the case of flowering, what happens is कि auxin has the role in flowering in some plants such as lychee and pineapple. Of roots, तो हमें high concentration of auxin चाहिए. This will help to induce the roots in the plant, root development. In the case of flowering, what happens is कि auxin has the role in flowering in some plants such as lychee and pineapple. In these two plants, it causes flowering. But ये है कि if we spray a lot of auxin on a plant, it will stop flowering. जैसे there are plants like lettuce. Okay. Now in this case, the only commercial need of this plant's part is the leaf. और leaf के लिए we have to prevent flowering. So for this, अगर हमने high concentration of auxin use कर दिया, it will stop flowering. Now coming to parthenocarpy. Parthenocarpy means parthenocarpy में what happens is that if we want to develop, let's say, seedless variety of fruits or vegetables, जैसे seedless variety of tomato or banana, तो हम parthenocarpy use करते हैं. और parthenocarpy को successful करने के लिए the best thing is auxin. So auxin is used, seedless variety of seedless variety of what? Of tomato and banana. सीडलेस वैरायटी ऑफ टमेटो एंड बनाना हम प्रोड्यूस कर सकते हैं ऑक्सीन के स्प्रे से। वीडीसाइड, ना ऑक्सीन जो है सर्टेन ऑक्सीन सच एज 2,4-D और 2,4,5-D को यूज करा जाता है एज अ वीडीसाइड टू किल वीड्स एज वेल एज टू किल ब्रॉड लीव्ड डाइकॉट्स। ओके, सो नाउ इसका एक एग्जांपल है कि व्हेन वाज इट यूज्ड, इट हिस्टोरिकल एग्जांपल है। So the next phytohormone that we are going to talk about is gibberellin. So gibberellin भी एक plant growth promoter है. It's a plant growth promoter. Now gibberellin जो है it is this the discovery जो है discovery is done by E. Kurosawa, who was a Japanese scientist. So how was it discovered? उसके डिस्कवरी में ये हुआ था कि जापान हैज राइस एज their staple crop. So जो सारे crops थे, some of the plants in that crop, they grew very thin, taller than the other plants. Let's say these are the other plants. These were the normal plants, and this was a thin, weak. It was a thin, weak, tall, and sterile plant. So this इसे एक disease कहते हैं वहाँ पे, which is known as Bacane, जिसका मतलब है foolish seedling disease. Now, what did E. Kurosawa do? That what he did was he took the plant and उसमें उन्होंने discover किया कि एक तरह का fungi था, fungus, that is Gibberella fujikuroi. Now Gibberella fujikuroi से जब उन्होंने extract निकाला और normal plants में ट्रांसफर करा, उसका रिजल्ट ये था कि जो नॉर्मल प्लांट्स थे, even they started growing too tall and becoming the same and showing the same symptoms as the foolish seedlings in this case. So next was Yabuta. Yabuta was another scientist जिन्होंने इसे extraction करा था. Extraction of this compound, that is gibberellin, और term भी उन्होंने दिया था gibberellin. Okay, so what are the physiological functions of gibberellin? Let's discuss that. Physiological functions. Now physiological function, क्योंकि ये plant growth promoter है, so growth करवाएगा. So the first thing is growth, that is it will cause the growth of stem and leaves. Okay, the second method is by bolting. Now bolting एक प्रोसेस है जो रोजेट प्लांट्स में होता है. Now what is a rosette? Imagine cabbage, and then cabbage में there are layers and layers of leaves. all wound around an axis. So this is called the rosette condition. Rosette condition में क्या होता है कि the growth of the internodes is shortened, जिसकी वजह से the plant becomes, let's say, it develops leaves something like this. But what does gibberellic that is gibberellin do? Gibberellin को हम GA से भी denote करते हैं, and it has 3 varieties, that is GA1, GA2 and GA3. Okay, so what it does is it promotes elongation of the internodes. And इस process को हम bolting कहते हैं. It occurs in, let's say, plants like beet, cabbage, etc. Okay. The last and the most important one is seed germination. Okay. Now seed germination में how does gibberellic acid work is it works by activating hydrolytic enzymes. Okay, hydrolytic enzymes activated. जैसे ही हाइड्रोलाइटिक एंजाइम्स एक्टिवेटेड होंगे सच एज अमाइलेजेस, वो क्या करेंगे कि वो जो सीड है, लेट्स से दिस इज द सीड के अंदर जो स्टोर्ड न्यूट्रिएंट्स हैं उन्हें वो मोबिलाइज कर देंगे, एंड दिस न्यूट्रिएंट विल बी यूज्ड फॉर द ग्रोथ ऑफ द प्लांट. ओके, सो नाउ वी विल टॉक अबाउट द यूजेस ऑफ this plant hormone. The first use is the growth of fruit. It also helps in increase in malt. It causes increase yield in sugarcane, plus gibberellic acid also causes quicker maturity. Okay, so let's discuss individually ki what are the actual things that are happening. Growth of fruit में what happens is gibberellic acid वो fruit के size को increase करता है. जैसे example is apple, Pyrus malus, apple का scientific name. So apple के case में what gibberellic acid does is कि वो apple का size plus उसका length जो है वो increase कर देगा. In short वो productivity increase कर रहा है. That could be used in a commercial way. Second is increase in malt. जो brewing industries होती हैं जो let's say alcohol या malted whiskey या something malted products produce करती हैं. So if the plant is treated with gibberellic acid, the malting process जो होती है malt का production that increases manifold. Okay, and increase in sugarcane yield. Sugarcane के हमने इस केस में देखा था कि as soon as gibberellic acid is added to the crop, उसकी length बहुत बढ़ जाती है, and that is an advantage in the case of sugarcane. Why? Because जैसे ही the length of the plant will increase in the case of sugarcane, it will translate to increase in the sucrose content. Okay, so it will lead to an increase in the sucrose content of the plant, जिसकी वजह से productivity will again increase in productivity. Yes. The last one is quick maturity. कुछ plants होते हैं जैसे pine cones, जो meal pine cones होते हैं, what it does, जिब्रेलिक एसिड से अगर हम उन्हें ट्रीट करें, so उनकी जो mature होने का टाइम है, it will gradually decrease. So इसकी वजह से you will get more number of let's say seed formation बहुत जल्दी हो जाएगा, या more number of seedlings you will get in a less time. जैसे इसका pine cones, तो you can get commercial example would be चिलगोजा, और pine nuts को प्रोड्यूस करने के लिए we can use this ताकि maturity जल्दी हो जाए. So you can derive the seeds from these plants. Okay. Now we move to the third phytohormone, which is known as cytokinin. Now cytokinin उसके नेम से ही पता चल रहा है, it is going to promote cytokinesis. Okay, going to promote here cytokinesis. So इसकी जो डिस्कवरी थी, it was done by Skoog and his colleagues. Now Skoog ने क्या ऑब्जर्व करा कि if they had a callus, now callus क्या होता है? Callus जो होता है, it's an undifferentiated mass of cells. Usually जब कोई भी plant cell या animal cell को कल्चर करते हैं, and if it's not it has not differentiated, तो हम उसे callus के नाम से जानते हैं. So जब एक callus था, and callus के ग्रोथ के लिए Skoog ऑब्जर्व कि यूजुअली उसमें ऑक्सीन चाहिए था, प्लस उसमें आइदर ईस्ट एक्सट्रैक्ट चाहिए था, या उसमें चाहिए था कोकोनट मिल्क. ना इससे डिड्यूस हुआ कि जो ईस्ट एक्सट्रैक्ट या कोकोनट मिल्क में देयर इज सम सॉर्ट ऑफ केमिकल दैट इज वेरी इंपॉर्टेंट फॉर द डेवलपमेंट ऑफ द कैलस इनटू अ प्रॉपर प्लांट. इसका द फर्स्ट टाइम समवन फाउंड आउट cytokinin was in an autoclaved herring sperm. Okay, an autoclaved herring sperm में they found out a substance जिससे आज हम cytokinin के नाम से जानते हैं. So next what they found out was कि unripe या जो पके ना हो, अनराइप मेज के जो कर्नल्स होते हैं, जब उनसे भी एक्सट्रैक्ट करा गया, देयर वाज अ सब्सटेंस व्हिच वाज एक्सट्रैक्टेड व्हिच वाज नोन एज zeatin. These are all forms of the same cytokinin, and इस केस में उसे काइनेटिन के नाम से जानते थे, काइनेटिन. ओके, zeatin एंड सेम चीज कोकोनट मिल्क के साथ भी हुई. जब इस zeatin या ऊपर जो काइनेटिन है उसे किसी प्लांट में इंट्रोड्यूस करा गया, तो प्लांट की जो ग्रोथ रेट है वो इंक्रीज हो गई. ओके, सो इससे पता चला कि इट इज वेरी इंपॉर्टेंट बिकॉज इट इज कॉजिंग सम सॉर्ट ऑफ लेट्स से एक्सेलरेशन इन द ग्रोथ ऑफ द प्लांट. और उसका रीजन क्या था? उसका रीजन ये था कि साइटोकाइनिन जो है इट कैन कॉज लेट्स से सेल डिविजन इवन इन द परमानेंट सेल्स. ओके, परमानेंट सेल्स। उसके अलावा इसके दूसरे फंक्शंस क्या हैं, इसके फिजियोलॉजिकल रोल क्या है, लेट अस डिस्कस दैट। ओके, सो इसके जो दूसरे फंक्शंस हैं, okay, it will also cause growth of shoot, plus it will stop apical dominance. Okay. So cell division जो है it's as such very important plant के growth के लिए. So इससे पता चलता है कि if we need to find out cytokinin, it will obviously be found in the growing parts of the plant. Okay, वहाँ पे you are likely to find cytokinin. So it will cause cell division even in permanent cells, जो differentiated cells हैं उनमें भी, as in the case of like let's say the callus. It will definitely cause the growth of shoot, लेकिन इसका कोई रोल नहीं होगा रूट के डेवलपमेंट में. प्लस इट विल स्टॉप एपिकल डोमिनेंस. एपिकल डोमिनेंस जो है इट वाज अ फेनोमेना जो हमने पढ़ा था किसमें, ऑक्सिन में. सो इन शॉर्ट इट विल वर्क एंटागोनिस्टिक या ऑपोजिट टू द एक्शन ऑफ ऑक्सिन, और एपिकल डोमिनेंस वो कम करेगा. एपिकल डोमिनेंस कम करने से क्या होगा, लेटरल ग्रोथ सीन होगी यहाँ पे, लेटरल ग्रोथ. ओके, so whenever we are culturing let's say a tissue, there are three conditions जो हो सकती है कि अगर we have what we have with us auxin, or equal quantity के we are using here cytokinin. It will cause the callus to remain as it is. Okay, so लेकिन अगर जैसे ही हमने ऑक्सिन की क्वांटिटी को इंक्रीज कर दिया साइटोकाइनिन से, it will cause root elongation या रूट का डेवलपमेंट will occur. और if हमने साइटोकाइनिन की क्वांटिटी को इंक्रीज कर दिया, so it will lead to the increase in development of the shoot. Okay, इसके अलावा there is another role, functional aspect, that is ये senescence को delay करेगा. Senescence is the old age or aging of the plant, so it will delay senescence. तो fruit या लेट्स से फ्लावर प्लांट पे ज्यादा टाइम के लिए रह सकते हैं इसके हेल्प से. नाउ इसकी यूजेस कहाँ पे होते हैं, इन डे टू डे लाइफ और इन इंडस्ट्री. द फर्स्ट इज क्योंकि ये सेनेसेंस के रोल को कम करता है, सो इट इज यूज्ड इन ओवरकमिंग senescence here. Okay, overcoming senescence, plus it is used as जैसे हमने यहाँ पे डिस्कस करा कि it is used for tissue culturing, क्योंकि without cytokinin जो root medium है it will not form or elongate. So this was about cytokinin. Let's now talk about another hormone. Now it is noteworthy here कि Skoog and there's another scientist who is named Murashige, a Japanese scientist. So they developed a media जो plant growth के लिए, which is very essential. So most plant growth या plant cell culture जब हम करते हैं, so we use a media which is known as MS media or Skoog and Murashige and Skoog media. Okay.